Hello, everyone. My name is Veronica Hood, and I'm the Scientific Director for DSF. I'm really sorry that I couldn't be there in person to greet all of you face to face. I hope to see you at a future event, and I hope that you can reach out to me at any time if you have questions about anything I present here today or a question that arises during your journey in the future. I've listed my email address here on the slide, and I'm also available if you ask a question in the private Facebook caregiver group. You can tag me there, and I'm happy to respond there as well. Today, I'm going to give a brief overview of the clinical picture of Dravet syndrome, and then I'm going to focus in a little bit more on how research has progressed and talk about some of the exciting therapies that are in development right now, as well as in clinical trials for patients with Dravet syndrome. I wanted to start out with this graphic here that you've probably already seen once today during the DSF programs talk, but we'll zoom in just a little closer and just focus in on some of the incredible progress that's happened in the overall field of research for Dravet syndrome. Dravet syndrome was first described in 1978 by Dr. Charlotte Dravet. At the time, it was called SMEI, or severe myoclonic epilepsy of infancy. But about a decade later, they decided to rename it Dravet syndrome so that it didn't appear that it was limited to infancy or limited to a specific seizure type, since we all know it's much broader than those terms. It wasn't until the early 2000s that scientists found the genetic variations in SCN1A were really highly associated with patients that had a diagnosis of Dravet syndrome. And at this time, families really thought that this discovery was going to launch research forward very quickly. However, they were discouraged when they saw that things really weren't progressing that rapidly. So here in this graphic, the bars on the top show the number of scientific publications each year related to Dravet syndrome. And so you can see in those first years that while there were some and there was a slight increase, it wasn't drastic at the time. And it was coming from the same groups of researchers. At the time, there was also very limited amounts of funding coming from the National Institutes of Health towards research related to Dravet syndrome. And so even by 2009, the NIH was only directing $2.2 million a year towards the research related to Dravet syndrome. And so when the foundation was established in 2009, it was really families coming together saying, how can we help spur the field of research on? Can we fund some small pilot grants that might help researchers then apply for larger NIH funding? And what you saw in the years following DSF's establishment was a lot of robust growth in the field, more and more scientists coming in to focus on Dravet syndrome, a drastic increase in the number of yearly publications, as well as big increases in the amount of money that NIH was spending towards projects related to Dravet syndrome. I think since around 2015, we've continued to see over $10 million a year directed towards Dravet research from the NIH. In addition to those measures, you also see here noted that in 2018 and 2020, there were 3 medications that received FDA approval specifically for Dravet syndrome, which was a very big deal since, even in the early 2000s, there were no medications specifically for treatment of Dravet syndrome. And so, with that mission that DSF had to focus on research that was primarily to fundraise and then to fund grants. And to date, the foundation has been able to direct over $6.7 million to research. And by the end of 2023, that number should be even higher as we're going through our grant review process right now. So I'm going to transition and first do a little bit of background on the clinical picture of Dravet syndrome. Again, I'm not a clinician, and so this will be a brief overview. If you're interested and want to know more about the basics of Dravet syndrome, we have a lot of great talks and webinars on the website from expert clinicians giving this overview, and I'd be happy if you reached out to direct you to a few of those. So most of you know that Dravet syndrome onsets generally within the first year of life in an otherwise very typically developing infant. They present with an initial seizure, which can either be a partial seizure or partial progressing to generalized, usually tonic or tonic-clonic. We also know that over time, additional seizure types might present that are different than those initial seizures. And so that's actually part of what I'm showing here in the graphic to the right. I'm not going to go into details. This is obviously a very complicated graph, but what you're seeing is that as patients age, so across the bottom axis here, as patients get older, the types of seizures that they have, the presentation, and some of the risks associated with seizures can also change. And so, as all of you know, this is one of the complex things that makes Dravet syndrome incredibly difficult to treat, because you're constantly having to make the decision about whether this is a medication that's causing side effects, a medication that's worsening seizures, or is this just a change in your child as they age. And so, as all of you know, that makes decisions very, very complex for both parents and for medical professionals. We also know that there are common triggers for seizures in Dravet syndrome. And again, these can also change over time, but the most common one is fever or temperature sensitivity. And that's why we see a lot of patients having that initial onset of seizures following their very first round of vaccines as an infant, because so often they'll spike a very low grade temp, which can then lead to the onset of that first seizure. We know vaccines don't cause Dravet syndrome. We know this is an underlying genetic cause. And we also know that patients that don't receive vaccines still end up, can still onset with Dravet syndrome. Dravet syndrome, though, again, as I'm speaking to a room of parents that I know are also experts in this disorder, so I don't have to tell you that Dravet syndrome is more than just seizures. Dravet syndrome is actually classified as something called a developmental and epileptic encephalopathy, or DEE. And that means that in addition to the very difficult to treat seizures that don't respond well to medications, that there are also impacts on neurodevelopment. And those impacts can be both due to burden of seizures as well as the underlying cause of the syndrome. Some of the non-seizure symptoms in Dravet syndrome include those neurodevelopmental impacts, such as delayed or reduced cognition, impacts on motor skills and development, delayed or disrupted speech, as well as very significant behavior challenges that families often see rise to be a bigger concern than the seizures as patients get older. There can be really incredible difficulties with sleep. And in addition to the motor impairments that can happen developmentally, we also see some motor impairments, particularly related to gait and how patients walk and move around, especially as they hit adolescence and move into adulthood. And so again, here I've got this very complex chart to the right, but instead of seizures this time, it's showing how some of these other symptoms of Dravet syndrome, these other comorbidities, can evolve and change and accumulate over time. Again, as all of you know, making this a very difficult syndrome to predict. A study by DSF in 2015 found the prevalence of Dravet syndrome to be 1 in 15,700 live births. So this study was done in the US, but we know that Dravet syndrome knows no geographic or ethnic bounds. And so this represents a similar incidence around the world. This was a really important study because before this, the previous incidents had been the previous incidence had been estimated at 1 in 40,000. And so this study showed that Dravet syndrome is actually a lot more prevalent than was previously thought. And so that converts to estimating around 17,000 and 19,000 patients just in the US alone. Excuse me. Unfortunately, we also know that premature mortality is a big risk factor for patients with Dravet syndrome, and it impacts about 10 to 20% of individuals. The most common causes of this are complications following status epilepticus or seizure-related injuries. But the leading cause of death in Dravet syndrome is sudden unexpected death in epilepsy, or SUDEP. We're still learning a lot about the causes of SUDEP. It's a very difficult phenomenon to study, but researchers believe it has a lot to do with respiratory and cardiac suppression following a seizure. Most often, SUDEP occurs after an unwitnessed seizure. It's very common to happen while patients are sleeping. And so right now, while we're still learning about SUDEP, one of the largest preventative measures you can take is to be really aware that this is a risk and to monitor patients, particularly during sleep. And so you've probably already heard today about the patient assistant grants that DSF offers. Those can help you if you're interested in purchasing seizure monitoring devices. We have a few webinars that go into more detail about those devices, and you can also visit the Danny Did Foundation website, and they'll break down more about the devices. Again, we're learning more and more. Exciting research just came out that is getting closer to understanding what some of the causes are for SUDEP, and hopefully that can lead to real interventions that can be done to help prevent further loss from SUDEP. All right, so we know that Dravet syndrome is a monogenic disorder. So that means it's caused primarily by mutations in a single gene. I mentioned before the SCN1A gene. So SCN1A codes for something called NAV1.1. And this is a really important sodium channel for cells in the brain to be able to communicate. We know that variants in SCN1A that cause Dravet syndrome lead to something called haploinsufficiency. The way that I remember easily what this word means is that haplo is half and insufficiency is not sufficient. So when you only have half of the amount of SCN1A in the brain, it's just not sufficient for cells to function the way that they should. We know that anywhere between 85 and I think the most recent estimates suggested over 95% of patients have mutations in SCN1A. They're not on the exact same place, and they're varied types of mutations. There have actually been over 1,700 variants reported in the literature so far, again, making this a little bit more of a complex disorder. Although all of the variants are predicted to, again, cause that loss of function and haploinsufficiency if they lead to Dravet syndrome. And so here's just a depiction of how everyone should have 2 healthy copies of the SCN1A gene. So this is your DNA. It makes a strand of something called RNA, which is basically the cell jotting down the recipe for the protein that it needs to make. In this case, it's the NAV1.1 sodium channel. The cell can then use that RNA message to make the sodium channel, and those sodium channels then find themselves along the membrane of a neuron in the brain, and allows that cell to bring sodium ions in and out, and that helps the cells set up an electrical charge so they can communicate with surrounding cells. Now, when a patient with Dravet syndrome has a mutation in one copy of the gene, they no longer make half the amount of healthy sodium channels as they should. And so now these cells only have that half of that, and they can no longer bring sodium in and out of the cells efficiently. And so they're not able to communicate as well, which is part of what can lead to the uncontrolled electrical activity that leads to a seizure. Now, not every single SCN1A mutation causes Dravet syndrome. So we have first a whole spectrum of disorders that can be caused by mutations or variants in the SCN1A gene, anywhere from the more mild end of the spectrum where you have familial hemiplegic migraines, all the way down to the more severe end of the spectrum where you have disorders like Dravet syndrome, and then some newly described disorders called gain of function DEEs, which instead of the loss of function that causes the haploinsufficiency, you have a gain of function, so a mutation that causes the sodium channel to not lose its function, but to do something additional that it shouldn't. And so that can lead to these gain of function DEEs. There's a few red flags about these because previously we didn't understand them. A lot of these patients were diagnosed with Dravet syndrome. If your loved one had a seizure onset before the age of 3 months, that's sort of just the first red flag of, hey, let's look back at the genetic report again and think about if this could be gain of function. Now, there are certainly patients who definitely have Dravet syndrome and loss of function variants that have an earlier onset. So that's just a red flag that we should go back and look at it. It doesn't necessarily mean that your loved one has a gain of function. But the reason that it's really important to look at those gain of function mutations are that sometimes those patients can actually respond well to sodium channel blockers, which would normally be contraindicated for patients with Dravet syndrome. There's also some less severe forms of epilepsy syndromes that can also be caused by SCN1A mutations. And so some of those can even overlap with the mutations that cause Dravet syndrome, making it sometimes really difficult in early childhood to know if a patient is going to fall on the Dravet syndrome end of the spectrum or something more like GEFS plus. And then even within Dravet syndrome, we know that no two patients are alike. There's a broad range of how patients can present. And it's not as simple as, well, this patient's more mild and this patient's more severe. I think about it sort of as a tuning slider that I've shown here, where each symptom can really be slightly different in each patient. And so just knowing that it's difficult in Dravet syndrome to directly compare to other patients. And the best thing to do is to be, optimistic about your child's future. We have better treatments than we've had ever before, and patients are getting diagnosed really early, meaning they're not being exposed to drugs that they shouldn't be on. But also being aware of what the challenges are commonly in this disorder and being prepared for the therapies and supports that your child might need over time. So just as I just mentioned, this chart here to the right shows sort of a snapshot in 2017 of the population of patients with Dravet syndrome. And at that time, if you looked at patients that were adults or adolescents, you could see that they had primarily been diagnosed at slightly older ages. So at the earliest, 1 to 2 years, but if you look at adults, they were mostly all diagnosed when they were 4 years or older. But if you go back and look at the same time at patients that were infants or in very early preschool, you were seeing that the majority of those patients were getting diagnosed within the first year of life. So we know that as time has progressed, clinicians have gotten so much better at quickly diagnosing Dravet syndrome. And that's because of a few reasons. First is that there's just an increased awareness of Dravet syndrome and other rare diseases, and clinicians are getting better at recognizing those and being more abreast of what those symptoms might look like. The other thing is the discovery of the gene allowed for a genetic test to be able to help point clinicians in the right direction. Now, something I haven't mentioned yet is that while most patients do have mutations in SCN1A, there are a few other genes that have been associated with Dravet syndrome more rarely, and a negative genetic report with a report that has no genetic findings, that patient can still be diagnosed with Dravet syndrome if all of their symptoms really fit the clinical picture. But like I said, the majority of patients do have those mutations. And so the advent of genetic testing and then the increased accessibility over the years for genetic panels has really decreased the time to diagnosis, which I really think is going to make a big difference in the long-term outcomes for many patients with this disorder. Importantly, that diagnosis means that we can at least use a little bit more directed approach to choosing medications, to choosing anti-seizure medications. In 2022, DSF helped to fund and participate in an international consensus study that brought together over 20 clinicians internationally to give their expert advice and expert experience on which medications have worked well in the hundreds of patients with Dravet syndrome that they treat. And so they came up with this chart of what they would most commonly choose first, second, third, fourth to either use as a primary medication or to add on to an existing medication regimen. Every single patient is different, as I've already mentioned, and that also extends to how they respond to medications. And so it's important to know that this is a guide and a starting place, but what works for one patient may differ slightly for another. All right, so again, I said that overview of clinical stuff would be fairly brief. Again, I can direct you to more if you have more questions about some of those basics, but I'm going to switch over to talking a little bit about the research progress that's happened and more about the role DSF has played. I think I've already touched on that a little bit just in talking about how things have changed with the clinical picture. But what has DSF done, if we reflect back on this graph, that really helped to impact those publication numbers and funding numbers? And so the way that DSF has approached this is primarily to fund pilot grants. So we fund grants that are anywhere from 75,000 up to 500,000 that last between 1 and 3 years for applicants. And those seem like really big numbers, but they're actually small compared to some of the NIH grants that generally give about 500,000 per year over the life of a grant. And so these are hopefully grants that help researchers spur a new research area or a new idea, get enough data that they can then go pursue a larger funding from somewhere like the NIH. We've worked with our scientific and medical advisory boards to be very strategic in the areas that we fund. We've kind of looked across all of the important areas shown in this graph to the right, and what we should really be thinking about is important to know and understand that could eventually improve treatments for patients. And so sometimes outside of our standing grant categories, we'll do special calls and we feel like there's a area that we really, that there's a hole in the field and we really need to know more about. So in 2019, we funded a grant to look more into a genetic therapeutic approach for Dravet syndrome. And this year, we're funding a grant that can try to help us understand more about sort of why patients with Dravet syndrome are such a spectrum. And is there something else in the background of their genetics that can help us understand why two patients that even have, the exact same mutation in SCN1A might have very different outcomes? And so now I'm just going to go over a few examples of some of the areas we've funded in and some of the work that's come out of that. We've seen such progression in the sort of research toolbox available to study Dravet syndrome. And a big component of that is having different models that you can study in a lab to ask questions, to test therapeutics before you move into humans. And so there are several mouse models, I think over 10 mouse models with 2 or 3 that are just very highly characterized and have been helpful in drug discovery so far. There's also zebrafish models, which again have been pivotal in drug discovery, and I'll talk about it just a little bit more in a moment. There's a newly developed rabbit model of Dravet syndrome that's going to allow researchers to understand hopefully more about SUDEP and some of the impacts of SCN1A mutations on the heart. And then there are also a variety of cell models, including induced pluripotent stem cells, which are created generally from skin cells or blood cells from a patient. And then we can turn them into stem cells, which then researchers can direct to become any other type of cell in the body. So they can create cardiac cells in a dish, they can create neurons in a dish, and it allows them to look at a cell type from a patient with the full background genetics of that patient. And so these types of resources exist in a lot of academic and for-profit research labs. But one thing DSF has noticed is that there's not an easily commercially available resource for iPSC-induced pluripotent stem cells from patients with Dravet syndrome. And so right now we actually have an effort going on to establish that. We're going to do 5 lines across varied SCN1A mutations. And it'll just involve a blood draw and providing medical information, and then we'll be able to bank it with a cell processing company that can then allow researchers to purchase it at a very low cost if they're interested in studying Dravet syndrome. If you're interested in participating in this effort, you can reach out to me and I can give you more information. We just started recruiting right now. All right. So in addition to all of those exciting models of Dravet syndrome, a lot of those models we have then invested in drug discovery techniques. And so I mentioned zebrafish have been incredibly helpful for this. And so here are just some examples of how zebrafish have been used to screen really large libraries of drugs. And from these studies, we discovered how successful modulating the serotonin pathway might be for treating seizures and Dravet syndrome. And so most of you are probably familiar with fenfluramine or Fintepla, which works on the serotonin pathway. But these studies have discovered a few other serotonin modulating drugs that might be really efficacious for Dravet syndrome that work just slightly differently than fenfluramine. And so some of these are actually have gone from these initial studies in zebrafish and are currently in human clinical trials to understand if they could be beneficial. And I'll actually talk a little bit about these in a few moments. In addition to these large drug screens, zebrafish have also been useful in investigating metabolic targets. So researchers have been really intrigued by how successful the ketogenic diet is for Dravet syndrome, and we don't fully understand why it works so well to control seizures. But the ketogenic diet is very difficult to adhere to. And so wouldn't it be wonderful if we could figure out what inside the pathway from the ketogenic diet helps to reduce seizures? And could we develop an oral therapeutic or something like that patients then wouldn't have to follow the strenuous rules of the ketogenic diet, but could still see the benefits? And so zebrafish have been really helpful in trying to tease that apart as well. As I already mentioned, DSF has invested heavily in genetic therapeutic approaches. So here I'm just showing a few examples of how we've tried to fund research that can get around some of the challenges for development of a gene therapy for Dravet syndrome. SCN1A is a very, very large gene. And so for a haploinsufficiency, a traditional approach would just be to send a new copy of the gene in. But unfortunately, the delivery vectors that are currently the most effective and safest in humans are just too small to deliver that very large gene. And so could we use an alternative larger vector? Or are there other creative ways that researchers can come up with to get around the size problem? It's also very difficult to deliver a therapy to cells in the brain, which is a protected organ in your body. Things have to cross the blood-brain barrier to get to it. We also don't know if there's a distinct time where it would be most beneficial to intervene with a genetic therapy. Does it have to be early, or could this still be beneficial even in adult patients with Dravet syndrome? And so using mouse models, researchers are beginning to probe that and actually seeing promising things that even intervening in a mouse that's an adult long after the onset of their seizure symptoms, that genetic interventions may still be beneficial. And so could this inform future human studies? In addition to directly funding exciting research and helping those advancements along, DSF also works really hard to bring everyone together and to make sure that as researchers, as clinicians, as biotech and pharma, and as the government make important decisions about clinical care, about research directions, and about drug development, that the patient voice is also a really important part of that conversation, and that needs for patients and the symptoms that families would most like to see addressed are front of mind as we make progress forward on all these fronts. One of the ways that DSF does that is through holding a yearly research roundtable. We hold it just before the American Epilepsy Society meeting each year, and it's a great event to bring together a lot of these stakeholders to talk about progress and where we need to go. There's a few other examples of how Dravet syndrome has made sure that the patient and caregiver perspective is really front of mind when decisions are happening about research and drug development. There have been several studies that have directly highlighted the totality of impacts of Dravet syndrome on patients and families that extend beyond just the high medical needs for patients. In 2017, DSF actually specifically led a study that highlighted caregiver concerns across the large spectrum of medical and quality of life issues. And that's been a really important paper to be able to share with drug development companies and saying, hey, these are the symptoms. These are the things that are really important to families that you should be focusing on in your clinical studies. In addition to those efforts, in 2022, DSF held something called an externally led patient-focused drug development meeting. And this was a really important opportunity for the FDA to hear directly from patient families about the challenges that they face living every day with Dravet syndrome, as well as the huge unmet need for therapeutics. You can see the recording at the dravet-el-pfdd.org. And there was also a very important voice of the patient report that was developed after the meeting. It's housed on our website, but it's also linked on the FDA website. And the idea is that when the FDA is faced with a regulatory decision related to something for Dravet syndrome, that they will pull this report and then they can use directly the feedback from patient families to help impact their decisions about whether a clinical trial may go forward or a therapeutic is approved. All right, so all that research progress has really brought exciting things that are moving towards the clinic for patients. And so I'm just going to go over some of the trials that are ongoing, some of the opportunities for participation, and some of the things that I'm excited to see coming down the pipeline. You can find all the information that I'm about to tell you at dravetclinicaltrials.org. When you first land on the webpage, it's going to look like this. You can also find it from the mega menu under Research and Current Clinical Trials and Pipeline. When you first scroll down onto the page, the very first thing you'll see are a listing of all the actively enrolling clinical trials. So if you're interested in your loved one participating in a clinical study, this is a great place to come. I keep it actively updated. As soon as I have information from a study sponsor, I'm going to update the website with that accurate information. And then after you get past the actively enrolling studies, you'll come across a pipeline that looks really similar to what I've depicted here. That sort of gives you an overview of all the different therapies that are with a biopharmaceutical company right now and in different phases of development. So whether it's preclinical or in a phase of clinical studies, it'll be listed here. So I'm just going to go through these one by one with you and talk just a brief overview of them and talk about opportunities if there's a study. So the first experimental therapeutic here on the pipeline is soticlestat. It's also referred to as TAC-935, and this is being developed by Takeda. It's a novel mechanism, so it works different than any current anti-seizure medication that is approved right now. It actually targets the cholesterol pathway in the brain. Now, this isn't your dietary cholesterol, so it's not going to have any impacts on diet. Cholesterol is actually too big to cross the blood-brain barrier, and so any cholesterol in the brain is synthesized directly there in the brain. And so this therapeutic is thought to act on that pathway, and it's shown promising data in mouse models of seizure reduction. And then there have already been 2 phases of trials that have shown that it might be effective at seizure reduction in patients with Dravet syndrome. And right now there's a phase 3 trial enrolling. It's for patients aged 2 to 21, and it's a 14-week placebo-controlled study with opportunity to go into open label extension following that phase. The next several drugs, I've already touched on how we have really found that the serotonin pathway might be very important, a really targetable pathway that might greatly impact seizure activity in patients with Dravet syndrome. So we know that fenfluramine is already on the market. There's actually 4 different serotonin modulating drugs that are in the pipeline right now. And so I'm going to go through all 4 of those. There is a risk of something called serotonin syndrome if you're on too many serotonin modulating drugs at a time. So each of these, you wouldn't be on something like Fintepla and then be on a second serotonin modulating drug that I'm getting ready to go over right now. But we have heard directly from caregivers in the community that some patients might respond well to one serotonin modulating drug while another one doesn't work for them. And so I do think it still makes sense that there are so many of these sort of coming down the pipeline, because it may offer more options to patients, even though it's targeting the same pathway for one drug that possibly works better for a particular patient, or maybe a patient has less side effects from one serotonin modulating drug than another. So the first of these is lorcaserin. It's sponsored by ASI. This was previously marketed as a drug called Belviq that was used for weight loss. It was actually pulled from the market because patients were found to have a slight increased risk of developing cancer. It was a pretty small risk. I think it was 0.7%. So it was like 6.7% of patients on Belviq versus 6% of patients not on Belviq developed some form of cancer. So it was a real risk, and it probably wasn't a risk to balance against something that was being used for weight loss. But some patients with Dravet syndrome, before it was pulled from the market, had been using lorcaserin off label. So not as a weight loss drug, but as a drug to help control seizures. And so when it was pulled from the market, ASI worked with the community to try to help make sure no one lost access to that drug if they were using it as an anti-seizure therapy. And then right now they're running a phase 3 trial to better characterize the effectiveness of lorcaserin. And this is open to patients ages 2 and older, so all the way up to adults. And again, it's a 14-week placebo-controlled study. Next, we have clemizole, or EPX 100. This is developed by Epigenics Therapeutics, and this is the drug that came directly out of those zebrafish screens that were one of the earliest projects DSF helped to fund. This was one of the most effective drugs in the drug screen. And, it's gone through phase 1 safety studies, but we also know that clemizole used to be used as an antihistamine. And it was pulled from the market, not because of any sort of safety risk, but just because it wasn't a very effective antihistamine. There were newer antihistamines that worked better. But now we're finding that it might actually be a really important anti-seizure medication from all of those zebrafish studies. And so right now there's a study going on, again, ages 2+. So this can include adult patients with Dravet syndrome. Sorry about that. This can include adult patients with Dravet syndrome, and it's a 20-week placebo-controlled study. And then again, there's the opportunity for the open label extension so that you don't lose access to the medication while it's still going through trials. All right, another serotonin modulating drug, LP352. This is sponsored by Longboard Pharmaceuticals. They just completed enrollment in their phase 1b/2a trial. This was a basket trial, which means it wasn't just limited to patients with Dravet syndrome, but included a broad range of patients with different DEEs. And we expect to see the first readout of some of their data from that trial by the end of this year. And then our last serotonin modulating drug is BMB 101. This comes from Bright Minds Bio, and they've just finished up phase 1 studies, which were in healthy adult volunteers. And their results seemed really promising for this drug being fairly safe with its profile, and for them to understand where they might want to go with dosing as they move the next phase forward in patients. Okay, so now we're done with the serotonin modulating drugs, and I'm going to talk about BL001, which is sponsored by Bloom Sciences. And this is exciting because it's very different than any of the other therapies. It's a live biotherapeutic product targeting the gut microbiome. So this is really based off of studies of the ketogenic diet and how that changes the microbiome. And mouse data has suggested that if you can make similar microbiome changes in a mouse that's a model of epilepsy, that you can actually reduce their seizures without them being on the ketogenic diet by just modulating the gut microbiome. And so this company, Bloom Sciences, has decided to develop this product and see if they can replicate that in human patients. They've just finished phase 1 studies in healthy adults, and the data was promising for safety and dosing. And we continue to look forward to see when they might move into phase 2 and begin testing this in patients with Dravet syndrome. Another new therapeutic that's still in that preclinical phase, so they haven't moved into human studies yet, but it's a vitamin K analog that's been effective in animal models of epilepsy. And they just got a large grant last year, I believe, to really beef up their preclinical work and hopefully gather data that can support them moving into human studies. And then the last therapeutic is from IAMA or IAMA Therapeutics. It's IAMA 6, and this is something that is called a selective NKCC1 inhibitor. Targeting NKCC1 is interesting because not only has it showed efficacy in animal models that it might be able to reduce seizures, there's also been a lot of looking at NKCC1 in autism, and so it makes you suggest that maybe this could be more beneficial than just the treatment of seizures. Things are still really early. Like I said, they're just doing the testing in animal models, and we expect to see that data by the end of the year. And then hopefully by the end of this year, they'll also initiate those phase 1 studies that will test the therapeutic in healthy adult volunteers. All right, so those are all the more traditional medicines that are in development. We also know there's exciting genetic therapies that we're on the cusp of seeing, the first of which, STK001 from Stoke Therapeutics. This is something called an antisense oligonucleotide. It acts at the RNA level to increase the productivity of the healthy copy of SCN1A so that you can make more of those sodium channels that you're missing. It's a treatment that will have to be regularly administered via intrathecal injection. So that's similar to a lumbar puncture. Patients will likely need it every several months. They're still figuring out the exact dosing regimen, what that might be in current clinical studies. So far, things have been progressing really well in their phase 1b/2a study that assessed STK001 in patients aged 2 to 18. They've seen seizure reductions across all doses that they've used and all age groups. And just this year, we've seen sort of the first indications in a small subset of patients that have been on the therapy the longest, that there may be some positive impacts on communication, on motor function, and executive function, which suggests possible positive impacts on cognition. We hope to see additional data out from their study by the end of this year, and then hopefully next year we'll start to hear more about their plans for the phase 3 trial. Next is ETX101 from Encoded Therapeutics. This is more of a traditional genetic therapy, so it would just be a one-time administration. They deliver it with a viral vector, and it would be directly injected to the brain, but only a single dose would be needed. And like I said, it's difficult to replace the SCN1A gene, so researchers have had to come up with more creative ways. And so this actually targets how much the SCN1A gene expresses, and they can help the healthy copy, again, be more productive and increase levels of SCN1A that way. This company is getting really close to human clinical trials. They're working really hard with regulators right now, but they still have not begun any dosing in human patients as of yet. RT-101 from a company called Regel Therapeutics is, again, a very similar concept to what I've just described with Encoded. It would be a one-time therapy. It would be targeted to help increase expression from the healthy copy of the SCN1A gene. And Regel is currently working on preclinical data in mouse and cell models to see if this could be effective. And then the last company, many of you also may have heard of Tavard Biosciences. Tavard is Dravet spelled backwards. This company has been spearheaded by two caregivers to patients with Dravet syndrome who literally want to reverse Dravet. So they named the company Tavard. And they've been working on two different types of RNA-based therapeutics that could address Dravet syndrome at the RNA level, and again, hopefully increase expression of SCN1A. There's a few other study opportunities that are actively ongoing. Valtoco or intranasal diazepam, the rescue medication, the company Neurelis is currently trying to expand the indication to ages 2 to 5. So they're running a clinical study to see about safety and efficacy in this age range. Similarly, UCB has just begun the ORCID study to look at fenfluramine or Fintepla in patients below the age of 2. There's an important non-interventional study that's looking at biomarkers to help us better understand the brain and how it changes in response to medications. This is going on at Cook Children's Hospital in Fort Worth, Texas. It doesn't provide an intervention, but it uses some imaging techniques to help us better understand the brain. And then if you have a loved one with Dravet syndrome who is an adult, I encourage you to go on to dravetclinicaltrials.org and look at the listing for Dr. Danielle Andrade. She works really hard to better understand the progression of Dravet syndrome and the symptom presentation in adults with Dravet. And so I encourage you to connect with her and participate often remotely in her ongoing research. So I'll wrap my talk up by just encouraging you to get involved, get informed about the research that's happening, and then get going in whatever fits for you and your family, whether that means participating in a research study, whether it means raising awareness and advocating for Dravet syndrome, or doing fundraising efforts to help DSF as we fund research that's spurring things forward. Again, you can always reach me via email with questions. I'm so sorry again that I'm not there in person, but I hope everyone's enjoying their Day of Dravet event, and I hope that I get to see you at a future DSF event. Thank you.